Yes, 9 a.m. Pacific time. So again, I'm very happy and privileged to be here for another Global Immunotalk with my friend and co-organizer Carla Rodlin from Yale University, and another friend and great speaker today, Yasmine Belkaid from NIH. And so welcome everyone. We'll proceed for Carla to introduce Yasmine and then with the talk. Thank you so much.
Thank you so much, Elena, and welcome everybody. Hopefully you're joining us again, but in case you're new, we just want to briefly summarize our main goals, which is to make this available to all, so to benefit and inspire all of us in an egalitarian manner, and also to increase our opportunities to keep on learning without traveling. So welcome again to a new Global Immunotalk. We couldn't do this without our very gracious speakers that have accepted to present these talks. Please keep on checking this, as the list will be updated as we move forward. And I want to remind you that next week we will have Susan Kaik from the Salk Institute. Now, today it is my real pleasure to introduce our speaker, Yasmine Belkaid. Let me just stop sharing this, and hopefully, let's see, Yasmine, I think you're listening to us. She will soon turn on her video. Yasmine, we're super excited to welcome you to this talk. So just as a brief introduction, I want to share with you that Yasmine was born, raised, and educated in Algeria, where she earned her master's with honors in biochemistry at the University of Science and Technology Hourari Boumediene of Algiers. And I apologize for my mispronunciation. Now, what I want to tell you, Yasmine, you might not know that we have had colleagues from Algeria joining in the past. And I have an impression that we have many more today. So after her initial training in Algeria, Yasmine moved to France, to Paris, where she obtained her PhD at the Orsay University and Pasteur Institute, and then came to the US, to NIH, for her postdoctoral studies on the immune response to parasites. And it is there where she rose through the ranks to senior investigator and chief of the mucosal immunology section. Yasmine has other positions as well. I will not go much into that. She has received really prestigious awards and recognitions, but I think most importantly, those of us that are familiar with Yasmine's work know that she's a premier mucosal immunologist, and she really pushes the boundaries in this area. She has pioneered studies on the role of microbiota, both on tissue homeostasis and repair, as well as in the immunity to pathogens, and I think that's what we're going to learn more about today. So thank you, Yasmine, so much for accepting our invitation and for sharing your knowledge with us. Welcome.
Thank you very much. I mean, it's really a true pleasure for me to be invited to this seminar series, and really congratulations, Carla and Elena, to put this together. I'm actually very happy to be part of an initiative that allows access to science to part of the world in which it is actually sometimes hard to have access to science. So thank you very much for that.
Absolutely, absolutely. And so before we start, and as Selena reminded us last time, we have many young people listening to these talks, mostly in their 20s and early 30s. And so we would love to hear from you if there's anything as you look back in your scientific career and you transfer your own self to that time. What would you tell a younger Yasmine Belkaid?
So there's a lot of things I will tell myself, but I think there's one advice and one thing that I hope I knew when I was 25, which is I think to never hesitate to ask for help. Then it's not a sign of weakness. And I think this is something I felt I may have learned a bit late in life. So my advice to any of you, seek for help, seek for advice. And there are extraordinary people along the way that can help you navigate this complicated journey in science. But the first thing you have to do is probably put yourself in a situation of asking for help. So that's my advice.
Thank you so much. That's, yeah, very, very important. Thank you so much, Yasmine. And so we now let you start with your talk on microbiota control of host immunity. Thanks.
Thank you very much. So as I mentioned before, I'm really excited to have been invited to this lecture series. And I think I love the fact that it's reaching trainees all across the planet. And I think in the spirit of this, what I wanted to do is actually share with you slides of my lab. And the flags that you see on the bottom of these slides represent all the nationalities that are currently present in the laboratory. So I hope many of you across the planet can recognize your country and your flag. And I just wanted to say send a particular message to people in my own original country, Algeria, and hope that many trainees there can actually access to this talk and this lecture. So thank you very much for joining today and to share with us the science that we are doing. So one thing that I also wanted to say is that this lecture is not really the right platform to discuss unpublished data and preliminary data. So for the people that may be familiar with my work and with the work we do in the laboratory, I apologize, it would be mostly published data. What I'm going to try to do is actually place the work of the laboratory in the framework of the broader question that we're trying to address and how we're actually going forward with some of these ongoing questions. So I would like to first start to the fact that many of us, including my laboratory, is trying to understand the factors that control the immune system. And one thing that we need to remember is that we used to have a relatively naive perception of the immune system. And until relatively recently, we saw the immune system as an attack system, which function was really in place to eliminate the invading agent, the microbe. So it was this very war imagery around the immune system. But I think what has really become clear over the last 20, 15 years is that the immune system is way more complex than that. And what the immune system is really is a rheostat of host homeostasis that is really able to allow the system to adapt to extraordinary challenges and to restore balances of the host. It is really a motile system that bridges all the other systems and allows to restore homeostasis. What has also become clear over the last few years is that the immune system is able to utilize constitutive elements such as the microbiota, diet, environmental triggers, but also pathogen in some cases to reach education of the immune system to reach its optimal fitness and its optimal resilience. So under physiological condition, the immune system is really this massive system that allows the restoration and the maintenance of tissue and host homeostasis. So if we actually approach the immune system that way, we may start to think about disease, and in particular chronic inflammatory disease, as a point of no return for the immune system ability to adapt. So we have actually, in many parts of the world, reached a state of disbalance in which those environmental triggers have now been dysregulated. The kind of microbe that colonize our surfaces have clearly changed. The nutrition that we have can actually be irregular and actually not healthy in many countries, especially high-income countries. We are exposed to pollutions that actually affect all the different tissues, in particular the lung. We change the kind of drug we take, and the kind of infection that we are exposed to are no longer the infection we co-evolve with, but are the one that we're actually acquiring as the one we're actually experiencing right now via travels and so forth. So really, we can start to think about all these inflammatory disorders that are actually plaguing the world today, maybe as a way of the immune system to just reach a point of no return and incapacity to really maintain this balance of the host. So in the context of this broad question, what the laboratory is trying to understand is actually the crosstalk between the microbiota and the immune system, how nutrition can actually influence the immune system both positively and negatively, and how infection educates the immune system, but also how certain infection can induce scar in the immune system, leading once again to a point of no return. And we're trying to do that in the context of tissue microenvironment, with the growing understanding that each compartment is really regulated in a way that is highly specific to the site and contains its own network of cells and its own system of immunoregulation. So I'm not going to discuss all of this today, but what I'm going to do is actually focus essentially on some of the crosstalk between the microbiota and the immune system, which is one of the aspects of the work we do in the laboratory. So our journey into the exploration of the role of the microbiota and immune system really started with two very talented fellows in the laboratory. Nicolas Bouladou was a postdoctoral fellow at the time, and Jason Hall was a graduate student from the UPenn partnership with the NIH. And what they were able to do in a very simple set of experiments is that if you treat an animal with an oral antibiotics, these animals were less able to develop IL-17 responses to an infection and less interferon gamma responses. And this led to the expansion of macrosporia in this specific example. So this was really a first hint that the microbiota could really act as an adjuvant of the immune system. And the downstream effect of this was, of course, enhanced control of pathogen. Around the same time, work by Dan Littman and others really highlighted, and actually Dan Littman in particular, revealed that defined microbes in the GI tract, such as SFB, for example, can push these Th17 responses. And the ability of these specific microbes to push Th17 responses was associated with enhanced responsiveness and control to C. rodentium infection. So this actually began the journey into the exploration of the microbiota as an adjuvant of the immune system, really pushing protective immunity against pathogen. But what has really become evident over the last few years is that this control is way more broad, and it is not just in the context of host-pathogen interaction. So the microbiota has now been shown by numerous investigators and numerous work across the planet to be a fundamental regulator of the immune system. And virtually all aspects of the immune system can be controlled by the microbiota, ranging from its development to its function. So for example, the microbiota can, as I mentioned before, promote the immune system in a way that promote antimicrobial defenses, both innate and adaptive responses. The microbiota can also, promote immunity to vaccine, and this is actually a developing area of research. Really exciting work that was actually done by numerous groups has actually now pointed to the microbiota as an adjuvant in the context of immunotherapy. But of course, this regulation of host microbiota interaction has now been shown to promote allergic responses, further the disease in the context of autoimmune disorders, and of course, accelerate inflammatory disorders. And one of the most studied, of course, would be the context of inflammatory bowel disease. So many of the work that has been done so far has linked the microbiota to the immune system function. But over the last few years in the laboratory, we became quite intrigued by another question, which is really how the immune system is able to recognize the microbiota. And what is really the mechanism that allow the initiation of this dialogue, both in health and disease? And right. So what we need to actually remember is most of what we really know about the immune system, and that's including myself as an immunologist, came from the exploration of inflammation and pathology. So beautiful work that was done by Genoway and Polymath Singers and others really pointed to the idea that in order to initiate immune responses, you needed some degree of inflammation, tissue damage, and aggression. And together, this actually was a system that engaged the immune system. The result of this engagement was the development of a class of immune responses that had for consequences, when well regulated, to control the invading agent. This class of immunity is also the one that we study and we characterize in the context of immunopathology. So this inflammatory class of immune responses is something that most of us have developed an understanding of and have actually begun really, I think we're at a stage of maturity of the field, in the field. But I think this really led us with this paradox. The microbiota is able to communicate with the immune system. So work that was done by Andrew McPherson and others a few years ago really highlighted that the microbiota was actually sensed and could actually lead to the development of antibody responses in the gut. Subsequently, work that was done by numerous people, including our groups, highlighted that the microbiota at all tissue barriers was actually able to induce T-cell responses, cognate T-cell responses. However, this class of immune responses is able to be induced and accumulate in the tissue in complete absence of inflammation. So this really, places in this puzzle and trying to really understand what are the mechanisms and the feature of this class of immunity that remain really poorly understood. And we refer to this class of immunity as homeostatic immunity, and we would like to propose then this form of immunity may be a dominant class of immunity in the body based on the enormous abundance of microbes that live at all barrier surfaces. So over the last few years in the laboratory, what we have done is ask several questions. I'm going to just travel with you in some of them. So we're very, very interested to try to understand the factors that allow the engagement of this dialogue between the microbiota and the host. What are the microbiota determinants, but also what are the host determinants that are able to initiate these responses and do so in absence of inflammation. I'm going to discuss with you some of the work we have done recently on the microbiota side, The host determinant is something that is more currently under development in the laboratory. But what we actually have spent a lot of time studying, and it's something I'm going to discuss during this talk, is really what are the unique properties and function of this class of immune responses that develop in absence of inflammation and can go back to the tissue without causing any kind of form of inflammation, and clearly without controlling the microbiota. So how those cells are wired, what are their function, has really occupied a lot of our time over the last few years. So this specific line of research, although we started to work in the GI tract, as many other people in the field, this specific line of research, in particular to try to track the antigen-specific response to the microbiota, started in laboratory when a very talented graduate student, Shruti Naik, who today has her own laboratory in NYU, joined the laboratory. And she was very inspired by the work of Julie Segre and Heidi Kong at the time, that had shown that if you looked at the skin microbiota of a human, the human is actually composed of a kaleidoscope of microbial community, very much defined by the unique landscape and ecological landscape of the different sites. So this was really the beginning of this survey of the high diversity of the skin microbiota. The microbiota in the skin lives in specific structures. It will be present in sebaceous glands, sweat glands. It's really very much embedded in the tissue and potentially in contact with cells that may be able to communicate with the immune system. So what Shruti was able to do, using various combinations of germ-free settings and antibiotic and so forth, was to show that very much like in the GI tract, the skin microbiota was able to control tissue immunity. And because of this control, the skin microbiota was able once again to act as an adjuvant and promote immunity to pathogens. So this actually led to a series of studies in the laboratory in which we utilized different kinds of microbes that we collected either from disease states in humans or in animals and wild animals, primates, and so forth. This microbe was subsequently applied at the surface of the skin in absence of any kind of abrasion or lesion, just painted at the surface of the skin. We looked at the immune signature that this microbe were able to impose on the skin and allow us to just create a collections of microbes able to impose specific immune signature on the tissue. And this actually told us two things. The first one is that very much that was shown in the GI tract by Kenya Honda and others, defined microbes have the ability to engage very specific immune cells. And the second thing, this actually now led us with a tool to allow us to look at the potential mechanism of association between one given microbe and the ability to engage one immune cell at a time. So the microbe that we spent a lot of time studying over the last few years, this is not the only one, but this is definitely the one we have spent most of our time, is Staphylococcus epidermidis. That is actually a microbe heavily present in all population. Actually, all of us carry this microbe. It is a microbe that can also colonize mice and can be found in some facilities. And if we looked at the surface, and if we apply the microbe at the surface of the skin of an animal, this is actually here, the skin of an animal that is raised in a clean facility. You can see that there is a very, can you see my, I hope you can see my arrow. Anyway, you have actually
yes, we can see.
Wonderful. You can see very few CD8 T cells accumulating in the skin at steady state. If you apply the microbe at the surface of the skin, there is this massive accumulation of CD8 T cells. These CD8 T cells have the ability to produce either IL-17 or interferon gamma. And as you can see, this production of this CD8 T cells occurs in complete absence of inflammation. The tissue is really unchanged. There is no neutrophil, there is no monocyte infiltration, there is no swelling of any sort. In the small survey that Shruti did at the time, she was able to show that this function appeared to be restricted to Staph epi and not other microbes we had actually tested at the time. So just to show you that this population of CD8 producing IL-17 is not an aberration in the context of this microbial association, if we utilize wild mice, so animals that had actually been colonized with a natural microbiota, and this was actually the kind of work that is currently done at the NIH by Barbara Rellman that has developed this really intriguing resource, an important resource for the community, we found that this population of CD8 producing IL-17 is naturally highly present. So clearly, animals in our own facility are now devoid of microbe able to push this important population of lymphocytes. So looking at this population of CD8 T cells, Alison Burt was a graduate student at the time who was sharing between my laboratory and the one of Julie Segre, she was co-mentored, was able to show that this ability of Staph epi to induce CD8 T cells was not a property of the entire species. For the people that may not be microbiologists there, and I think for the microbiologists it may seem very naive, but for the non-microbiologists, I just would like to highlight one point. Then when we think about the link between the microbe and the immune system, a species will never be a sufficient level of resolution. There is an extraordinary diversity of function among the species of bacteria. And in some cases, unique clades or unique strains may be the one engaging the immune system in a very specific way. What Alison was able to show is that this clade A20 was the one responsible for the induction of the CD8 T cell responses. And she was able to show that this clade A20 was in fact a feature of healthy volunteer skin, highly represented in human population. So very briefly on why and how the CD8 T cells are generated, we had very lucky partnership over the last few years with Michael Fischbach and Erin Chen in his laboratory. Erin Chen being a microbiologist and MD PhD in his laboratory, that was able to develop a novel approach, allowing to genetically manipulate Staph epi. And it was really powerful in the sense that her approach really allowed to manipulate primary isolates and really create a large collection of microbes that allow us to interrogate the factors that are responsible for the induction of different class of cells in the tissue. And by generating a large class of different mutants, she was able to show that specific mutants, although able to colonize the skin with absolutely no defect, were in fact defectuous in their ability to induce the CD8 responses. And to summarize this work, because actually this is published, actually it's actually in bioarchive at the moment, she was able to show, together with Nicolas Bouladou in the laboratory, then the way by which Staph epi is able to engage the immune system is via the expression of teichoic acid and lipoproteins that are able to engage in the host site TLR2 and dectin-1. This TLR2 is, of course, highly reminiscent of the work of Dennis Kasper and Mazmanian that actually highlighted the importance of these TLRs in the recognition of commensals in the GI tract, different commensals in the GI tract. So this actually recognition allowed the induction of those CD8 T cells in the host. But this, of course, led us with what I think is the most important question, which is, what is the point? What is actually the reasons of those CD8 T cells induction? So this came actually from the work of both Shruti in the laboratory and John Linham. So what Shruti and John were able to show is that the CD8 T cells induced by Staph epi have the ability to accumulate within the epidermal layer, which is very reminiscent of what people have actually shown in the context of tissue-resident memory cells directed against viruses, for example. So very much like this cell, like in the context of viral infection, the CD8 can actually go to the epidermis. Although at the difference of this other system, this is not an infectious process and there is no inflammatory process. We actually subsequently showed, this is not going to be shown here, that these cells are tissue resident, which means that they can accumulate in the tissue and persist for the long term. When they enter in the tissue, they have the ability to produce IL-17, and I would like to remind you that the antigen, the microbe, is still present. These cells are able to produce IL-17 and to communicate with keratinocytes in a way that promotes antimicrobial peptide production by keratinocytes. And this really enhances innate responses of the tissue, allowing the host to be better protected against subsequent infection. In the example that we published in the specific paper, this was against Candida infection. So this really allows adaptive immunity to a microbe, the commensals, to promote innate responses against subsequent infection, a phenomenon that can be referred as heterologous protection or immunity. But at the same time that the CD8 T cells are able to promote this fundamental process that is enhancement of immunity, what John was able to show is that very soon after an injury, and in this case it is a skin wound, the CD8 T cells are able to accumulate at the edge of the wound and create really a ring. And what John was able to show is that in addition of the antimicrobial defense program that is induced by these cells, the CD8 T cells are also able to promote tissue repair and accelerate this fundamental process that is required for host survival, that is a protection and restoration of tissue integrity. So this, of course, led us, once again, with numerous questions. I mean, some of them were, of course, what are the nature of the antigen that allow this kind of recognition? And second, how are those cells wired to be able to just promote such a distinct function of the tissue, ranging all the way from antimicrobial defenses to tissue repair? So one point that I wanted to actually highlight here, which is I think something that we need to all remember, is despite the fact that the microbiota contain putatively more antigen than in any organism that we can encounter, including self, pathogen, food-derived antigen, and allergen, we still understand very, very little about the nature of the antigen that the immune system is able to see in the context of host microbiota interaction. So we decided to first explore the nature of the antigen that could be potentially seen by the immune system in this specific example of staph host interaction. So what John was able to show is that, quite surprisingly, in the context of staph, the class of antigen that were seen by the immune system were unusual antigen in the sense that they were N-formylated peptide, which is of course a canonical feature of bacteria. This N-formylated peptide produced by staph can be presented in the mice by a non-classical MHC molecule, which is non-polymorphic H2M3 molecule. And this molecule has the ability to present this peptide and induce the CD8 responses. So it appears that in the specific example I have described to you, the ability of the immune system to communicate with this microbe seems to engage an ancient form of the immune system, which is really this non-classical MHC machinery, to recognize those unusual antigens that are N-formylated peptides. So this actually opened the possibility, which is of course ongoing at the moment and in collaboration with quite a few people, including Fiona and Federica, and it's a work that is done currently in the lab by Taylor and Sid. This actually opened the possibility that maybe one mode of communication between the microbiota and the immune system may be mediated via this ancient form of the immune system that is non-classical MHC molecules. And one point that may actually support this idea is that if we looked into the genome of numerous bacteria, these N-formylated peptides we found in staph are actually highly conserved in numerous bacterial species, both belonging to commensals or pathogens. So maybe one way by which the microbiota can communicate with the immune system is via the communication between highly conserved antigens that are present in the microbiota, presented by those really conserved molecules in the host genome. And I'm going to come back to this point later. So now coming back to the other question, which is what is how actually a cell is able to promote not only antimicrobial defenses, but also tissue repair. So Ollie Harrison, who was in the laboratory at the time and who has since then started his own group, was able to show that something quite intriguing was the phenotype of this cell was quite intriguing. In one hand, these CD8 T cells did express our gamma T, that is of course required for the production of IL-17 and other cytokines linked to these transcription factors. And this was of course the mechanism by which they could actually promote antimicrobial defenses. However, when he looked at the phenotype of these cells, he found that at the same time that they expressed our gamma T, the cells were able to express another transcription factor, GATA3, that is usually not expressed at high level in cells in the context of host defense. And this GATA3 transcription factor at high level is the one that usually mediates production of type 2 cytokines, something that has been shown to be linked to a phenomenon such as tissue repair. So this paradoxical coexistence of those two transcription factors is what Ollie was able to show was responsible for the ability of these cells to adapt to different kinds of challenges and express this functional plasticity. So to summarize the work then Ollie published, and one that is actually at the moment continuing in his own laboratory, what Ollie was able to show is that T cells generated against the microbiota, and this is in fact not just the CD8, this is also true for CD4, when they enter in the tissue, have a default program allowing them to produce IL-17 cytokines and our gamma T and so forth. And this is very much in line with the work that has been done by Dan Littman and others in the GI tract, showing that really the default program of a lot of commensal-specific T cells is around this our gamma T program. But at the same time, the cells have this unusual phenotype in the sense that they have this poised type 2 transcriptome. They do express messenger for IL-13, IL-5, and this really allows the cells in the context of injury, and we have actually done that in several injury processes, including the bite of insect or actually wound. In those cases, alarmin that will actually be triggered by, in the context of tissue damage, can act on the cells directly, and we found an IL-18 in particular can act directly on the T cells and now unleash these type 2 cytokines, and in particular IL-13 that we find to be required for this wound repair program. So these really tissue-resident memory commensal-specific T cells sitting in the tissue have this extraordinary ability to flip their program based on the kind of injury, allowing them truly to adapt to different forms of stressors and really representing a strong asset for the tissue in the context of adaptation to different environmental challenges. But of course, this has to be tightly regulated. And what Ollie was able to show is if you have a specific defect in immunoregulation in the tissue, via an impairment of the function of regulatory T cells in the skin, now the commensal-specific T cells can start to produce large amount of type 2 cytokines spontaneously, something that could actually underlie the etiology of allergic responses in the skin that are characterized by aberrant type 2 immunity. So really clearly trying to understand how these commensal-specific T cells are regulated in both health and disease will be important for us to try to understand how to regulate tissue in both settings. But this actually raises another point, which is the following. If we make the assumption that most of the antigen that can be seen by T cells could be microbiota specific, which would be likely the case based on the enormous amount of microbe that we're exposed to, this would actually imply then the vast majority of tissue-resident memory cells, and in particular those sitting in tissues colonized by the microbiota, are commensal specific. So really trying to understand the specificity of these cells, how they are regulated, I think is going to be a very important line of research for us to try to understand tissue-specific responses. But in this context, this actually opened the door in the laboratory to other lines of investigation. In particular, we became intrigued to try to track other antigen, and in particular canonical antigen expressed by the microbiota that can actually communicate between the immune system and the microbiota. And one class of antigen that we became quite intrigued by over the last few years are the antigen that derive from the metabolic pathway of vitamin B2. Indeed, one of the fundamental functions of the microbiota is to produce vitamins. And in this process, the microbiota is producing numerous metabolites. Some of them, in this specific example, will be the natural antigen for MAIT cells, that is a population of T cells that are highly abundant in humans. So these antigens are presented by the molecule MR1 and beautiful work that was done by numerous investigators, including Oliver Lantz and many others, have highlighted the presence of these cells, the systemic development, and so forth. These cells are highly abundant in humans. They can represent up to 10% of peripheral blood T cells, which is quite a high number. And they can represent up to 45% of all lymphocytes in the liver. So really extraordinarily high number of cells in humans. But mechanistically, I think it has remained challenging to explore the function of these cells because in mice, the cells are virtually absent. So this actually came to a surprise to Mike when he joined the laboratory and was intrigued to look at the presence of these cells in different tissue, and was able to show that very surprisingly, in his mouse colonies, MAIT cells that can be here detected via tetramer staining are in fact highly prevalent. So this was really contrasting with what we had found in the literature at the time, and it was really quite surprising. And this really led to the exploration of this population of cells and their function and their relationship with the microbiota. So what Mike was able to show is that these cells, as I mentioned before, are highly abundant within the skin compartment, but are virtually absent in germ-free mice. This population of lymphocyte is in fact the only one that is really almost exclusively depending on the microbiota. All the other population of lymphocytes tend to be decreased, but this is the most affected population in absence of microbe. And this reliance on microbiota has been actually previously published. So when Mike looked at the phenotype of these MAIT cells, he was able to show that in the skin, they tended to be mostly IL-17 producers. And in this specific colony of mice, he found that they could represent, in fact, the majority of the cells able to produce IL-17 in the tissue. So this was actually an intriguing observation that became more complex than we initially thought. And this actually was because the phenotype became highly variable, and it was highly depending on the cage effect. So animals that were genetically identical, or animals that came from different vendors, could have actually an extraordinary diversity in terms of frequency of MAIT cells in the skin compartment. And this actually is something that today we can acknowledge is usually due to the microbiota effect, especially this affecting animals that are genetically identical. This high variability in the MAIT cells is reminiscent of what is actually known in the human setting, in which MAIT cells frequency can be up to 10% of peripheral lymphocytes, virtually none. So this actually led to the exploration of the role of the microbiota, and especially different kind of microbiota in MAIT cell development. So the first thing that Mike did was to first survey the expansion of MAIT cells and other population of non-conventional T cells in different tissues, and was able to show that MAIT cells tend to accumulate in tissue, and in particular within the skin compartment, between the week 3 and 4 post-birth. And this actually supported the idea that microbes prior to this moment may be important for the accumulation of MAIT cells in tissues. So to address this question, what Mike did together with other people in the laboratory was to create a collection of microbes that were highly represented in the gut of colonies of mice that were able to generate MAIT cells. So then he took this mini collection of microbes that we will refer as neonate microbiota and seeded germ-free mice, neonate, with this population of microbes, and was able to show that just adding these 5 microbes was sufficient to increase the accumulation of MAIT cells in the tissue to level comparable to what would be found in SPF mice. But at this stage, Mike made also another intriguing observation, which is that if you take the same population of microbes and give them to animals that are adults, in this case, the MAIT cells are no longer able to develop and accumulate in the host. So this actually pointed to the idea that MAIT cells really are able to develop and seed the tissue, but this actually is dependent on a very specific window of opportunity that can no longer be actually obtained if we actually restore the right microbiota later in life. So to try to understand which mechanism was responsible for this accumulation, not surprisingly, of course, many of the microbes that Mike was able to actually look at express the machinery that will allow the production of the metabolite I referred to that are the natural antigen for MAIT cells. And indeed, when he actually associated neonate animal with a bacterial lactobacilli johnsonii that does not have this machinery, these MAIT cells are not able to accumulate. But if he now applies, associate the animal with P. mirabilis, this actually now allows the accumulation of these cells. So clearly, microbes enriched in this function are able to accumulate MAIT cells in the tissue and others do not. One thing that is intriguing and will remain to be explored further is that proteobacteria, which are really highly enriched in this function, tend to be also highly represented in early life in humans. So to which extent this actually may explain the role of these proteobacteria in shaping the immune system later on will be something quite intriguing to observe and to analyze. So the next step was to try to understand if the antigen alone, as opposed to the antigen plus other action that the microbe could do, will be important for this function. Mike did a very simple experiment where he took neonate animals and applied them with the ligand, and he did that using different routes. In fact, all of them worked, and found that if you do that in a neonate animal, just giving the antigen in absence of the microbe was sufficient to induce the accumulation of MAIT cells. But once again, the antigen in the adult mice was not able to promote the accumulation of MAIT cells. So in terms of interim conclusion, what I wanted to actually highlight based on the work of Mike is that there's clearly a window of opportunity for the development of MAIT cells. If the early life microbiota is enriched in a function that allows the production of those metabolites, MAIT cells can actually develop and accumulate in tissue, and more particularly within the skin compartment. Why the skin is something that we are now currently further exploring, but nonetheless, there is really this window of opportunity for development. If the microbiota is devoid of this fundamental function, MAIT cells are no longer able to develop, and this is not something that can actually be repaired by the subsequent addition of either the antigen or the right microbe. So this idea of window of opportunity is actually highly reminiscent from the work that has been done by numerous people in the world, highlighting the importance of this early life interaction with microbes and how this regulation of this early relationship in the microbiota and the immune system can have long-term consequences and in some cases lead to inflammatory disorders. So of course, to which extent what Mike uncovered here and the role of this metabolite and MAIT cell development early in life contributes to the etiology of some of those diseases could actually remain to be explored. But based on the extraordinary frequency of the cells in some human population, ranging up to 10% of peripheral lymphocyte and 45% in the liver, I think we can actually propose that indeed this early life exposure to those ligands may play a very important role in shaping the immune system for the long term. So the next set of questions was, of course, to try to understand what would be the function of MAIT cells. And as I mentioned before, although there is some hint that these cells can act in different settings, this line of research has been really complex because of the absence of animal model. So the goal of Mike was to try to see if you could push forward and actually push the function of MAIT cells and try to look at the physiological function of the cells under steady state condition. And for that, the first thing he did was to see if MAIT cells can actually be activated by exposure to new microbes after their development. can those cells, when they enter in the tissue, sense being actually be reactivated via the antigen recognition of new microbes. So to do this experiment, what Mike did is to allow the animal to develop MAIT cells, so they had the right metabolite early in life, and then apply the mice topically with staph epidermis. And he was able to show that by doing that, he can enhance the accumulation of MAIT cells in the tissue. This is not going to be shown here, but this accumulation does not rely on neogeneration of MAIT cells, but really the local proliferation of the cell. So once again, we turn out to our wonderful collaboration, Erin Chen and Michael Fischbach, and she generated some mutants and bacteria that were no longer able to produce the metabolite required for the MAIT cell activation, and showed that if you apply those microbes at the surface of the skin, they can colonize the skin, but they can no longer actually activate this process, and MAIT cells are no longer amplified by this association. So to try to see if antigen recognition was important for that, Mike developed an animal in which he can selectively remove MR1, which is of course the molecule able to produce the antigen in a very temporal manner by treating the animal with tamoxifen. And when he did that, he applied the animal with staph epi, remove MR1 expression, and you can see that now this accumulation of MAIT cells can actually be decreased by removal of the expression of the antigen-presenting cells, supporting the idea that antigen presentation locally is responsible for local accumulation of these cells. But this, of course, led us to the next question, which is the function of these cells. So what Mike did is to purify the cells after association with staph epi or in the context of association with the antigen, and looked at the gene signature that were expressed by this population of cells, and was able to show that once again, and this is something that seems to come every time we're looking at commensal-specific T cells, some of the dominant signatures were linked to tissue remodeling, tissue repair, and so forth. So this actually is something that seems to pop out in all the different experiments we do. And when we're looking at commensal-specific responses, we seem to actually propose at the moment, based on our work, that most of the responses we have tend to be by default aimed at tissue repair and tissue remodeling. So maybe one of the primary functions of the microbiota is to engage the immune system to this fundamental process that is tissue repair. So this, of course, is something that Mike now wanted to assess, and to which extent MAIT cells, and in particular enrichment in MAIT cells, can actually promote tissue repair. So to do that, what Mike did is a very simple experiment where he applied the animal topically with the ligand. So you can see here, actually a section of the skin of an animal in which you have the epidermis, and here you have few MAIT cells that will present at steady states. These cells tend to have this intriguing phenotype in which they are localized mostly within the dermis. Some of them appear to be in contact with keratinocytes, and something that is quite intriguing. But one thing that I always found very interesting, and actually is something that we're going to be following in different settings, is that all of the cells that are engaged by the microbiota seems to occupy specific macro niche. So for example, the MAIT cells tend to be within mostly the dermis and very likely close and likely to the epidermis. The CD8 T cells tend to be in the epidermis. The CD4 T cells tend to be within the dermis dominantly, and in the majority of the cases close to the hair follicle. So it's almost like each of them will occupy specific niche, and really trying to understand why this is the case is something that will be quite intriguing to explore over the next few years. So when Mike applied the ligand at the surface of the skin, you can see here that there is a massive accumulation of these MAIT cells. And this is actually just with few days of association with the ligand. When you look 2 weeks later, there is this massive accumulation of MAIT cells in the tissue. This is highly specific to MAIT cells and is not inducing any other population in the sites. So then looking at the ability of the tissue to repair, we utilize this model of punch biopsy in the back, in which you measure very precisely the ability of keratinocyte to proliferate and to migrate in the wound, in the bed of the wound, creating what has been referred as epidermal tongue. And you can see that if you apply the ligand at the surface of the skin 2 weeks prior to the punch biopsy, you can have an acceleration of this process, and there is a significant increase in the ability of the tissue to regenerate. So this actually proposed that MAIT cells, and it's not something that had been shown before, may at baseline, at least in steady state condition, and this will have to be proven, of course, in other settings, tend to be actually an important cell that can engage in this fundamental process of tissue repair. So the conclusion of this part is that clearly MAIT cells, and it's very likely the case for other population of cells, are really imprinted by very specific signal at a very specific developmental window. If the animals or the host is devoid of this fundamental function that allow the production of this metabolite, the ligand is not available to the in the thymus, the cells can no longer accumulate, leading to the tissue vulnerable to subsequent injury because there is a clear defect in MAIT cells accumulation. If, on the contrary, the host contains those beta-2 synthesizers and has this metabolic function, these MAIT cells can now develop and accumulate in the tissue. What we also found is that the survival of these cells and the ability of the cells to be maintained is under the control of IL-23 in the site. And then subsequently, in the context of a neo-exposure to a microbes, these cells utilize IL-1 and IL-18 to respond and to actually enhance this tissue repair program and really contribute to this fundamental process. So I think it will be very important over the next few years to continue to explore the function of this network of cells in the tissue, and in particular this fundamental relationship of commensal-specific T cells and tissue repair. So in conclusion, the microbiota is highly diverse, and I think this is something starting this journey that I felt a little bit overwhelmed, like many of you may have been at one point. The microbiota is so diverse, there is so many different antigens. But really, maybe one way by which the microbiota is able to communicate with the immune system, and maybe one way the immune system has evolved to communicate with the microbiota, is by using this simplified language, in which really highly conserved antigens that are present in the microbiota can engage those non-classical T cells that tend to recognize those highly conserved elements. So the examples I have given you here is about the MAIT cells and those non-classical restricted MHCI class 1 CD8 T cells. The work from Richard Blomberg actually highlighted a similar concept in the context of the GI tract and NKT cells. So to which extent this form of communication between those highly conserved antigen and those non-classical T cells represent the dominant form of communication in the microbiota and immune system, and the steady state condition remain, of course, to be further confirmed. But we could really propose that it's at least one of the important layers of this fundamental dialogue in the microbiota and immune system. So, of course, this class of immunity is, as I mentioned before, different from the one that we classically generate in the context of pathogen. It's a highly physiological form of immunity that has the ability to control numerous physiological functions of the tissue, ranging from antimicrobial defenses, tissue repair, and likely many other physiological functions that remain to be explored. So where are we going at the moment? So as I mentioned, there are still many, many questions to address. And I think one that we are very interested at the moment is really to try to understand the factors that control this balance between the ability of this homeostatic positive interaction microbiota and the one that become more aggressive. And I think one thing that we need to remember is numerous inflammatory settings can actually challenge our recognition of microbes and transform this healthy relationship with the microbiota as inflammatory. So indeed, over the last few years, work that we have done, but also many other people have done in numerous laboratories, highlighted that this ability to recognize the microbiota in a healthy way can be threatened and challenged by numerous settings. For example, defect in immunoregulation can actually transform this relationship as aggressive. Infection can actually now blur the distinction between the microbiota and the pathogen. A change in the diet itself can actually transform a relationship with the microbiota as inflammatory. And recent work that was done at the NIH by Pam Schwarzberg, and we were lucky to collaborate with her on that, highlighted in a model of primary deficiency, aberrant B-cell responses that are actually important in this specific disease tend to be mostly directed against the microbiota. So clearly trying to understand how those antigens are seen under steady state condition and regulate health and the tissue physiology, but also how those antigens now become agent of damage and inflammation is something that we're going to continue to explore over the next few years. And clearly we have a lot to do. So on that, I have to thank all the people that have done the work. I mentioned along the way the people that have left the laboratory and contributed to this work, but I also want to thank and give a particular thanks to my laboratory right now. They're all locked home. It is a hard time for many people across the world. So I would like to thank them very, very much for being here, for keeping their spirit up, and for being really an extraordinary group of people to work with. So I have been very privileged to work with many fantastic people over the years, and the current group is definitely one of them. So I have to also thank all the extraordinary collaborators. I have put many people there, not all of them were linked to this specific talk, but it's an opportunity to say thank you to all the wonderful people we have been lucky and we are lucky to work with across the planet. And I think this reminds me once again that actually science is all about collaboration and sharing. And I hope then this talk can be part of this sharing journey. Thank you very much for your attention.
Thank you so much, Yasmine, for such an excellent talk.
Yes, that was beautiful. Thank you so much. Really impressive, your research, Yasmine. Novel and elegant. Thank you so much. I enjoy it very much, and I'm sure this is a shared feeling by many that have been listening to you across the world. Yeah, thank you for the effort for being here and presenting. Yes. So before we close, as you know, questions are via Twitter. So just briefly reminding everyone that the steps for asking the questions via Twitter. So let's see if I can put this slide here. And we go through the very simple 3 steps to concentrate the questions for Yasmine. So first, make sure you search for the account Global IMMUNOTALK, and then in this account you will find the tweet that says ask questions for Dr. Yasmine Belkaid seminar here. And then you reply to that tweet with your question and mentioning the hashtag Global Immuno and the account Immuno Speaker, that is the account that Yasmine will be using to answer the questions. And so in this way, we centralize all the questions in one place. And thank you, Yasmine, again. And we will close just reminding you that next speaker is Dr. Sue Kaik from the Salk Institute, and we hope you join us again. So thank you, everyone.
Thanks, Yasmine.
Bye. Thank you.